Welcome back to Bite Size MCAT. In today's video, I want to walk through how I think it is beneficial to process through the glycolysis pathway. Time and time again, I see students brute memorizing glycolysis, but truly understanding how the carbons and phosphates move around in order to produce the various intermediates is necessary to apply this material on the MCAT or in your various biochemistry classes. So, first off, I don't want to scare you with this picture. We will go through all of it here shortly. But we will begin by taking a big picture of glycolysis, then discussing a strategy on how to think about glycolysis, before ending with walking through each mechanism step by step. Now, this video is not meant to be a complete replacement for understanding the biochemical structures of each intermediate, but it's supposed to give you a big overview about how to think about glycolysis so that learning the structures will become easier in the future. We will focus on counting carbons, counting phosphates, and then see how all of this applies to truly understand the glycolysis pathway. So, how do I want you to think about glycolysis? What has helped is instead of sitting down, just trying to brute force memorize everything, I have walked students through how to track both carbon and phosphate counts in order to understand what each enzyme is doing first. Obviously, memorization is going to be part of knowing glycolysis, but there are tips and tricks I will show you along the way to make it easier. Also, I want you to think about glycolysis in three phases. Most of the time, this is taught in two phases, which I think is great as well, but the middle phase here is quite simple to separate out by itself. This three-phase approach makes it far easier to digest and think about each enzyme rather than just memorizing 10 steps. So these three phases that we've seen on the previous side are investment, then cleavage, then payoff. So just think about it. First, we have to invest some energy into this process in the form of ATP, which we'll learn about. Then we're going to cleave this molecule in half, and then we're going to get some payoff. We're going to make some profit in the form of NADH and ATP. So our first phase is energy investment. The main thing I want you to remember is that our carbons stay the same, but we use energy. So at the beginning, we have a 6-carbon structure, and at the end, we have a 6-carbon structure, but with 2 phosphates. In this video, I'm going to denote each carbon as one of these little circles, and then I'm going to denote phosphates as these kind of aqua blue circles. So let's start by working through this. Okay, let's walk through this step by step. So first we have glucose. Remember that glycolysis is simply the breakdown of glucose. Let's just imagine that glucose just entered a cell. So what's to stop it from just leaving the same way it came through a transporter? Well, what we need to do is actually trap it. We have to stop it inside of the cell. How are we going to do that? Well, actually in glycolysis, what we do is we add a phosphate group. Again, think of this cyan circle as a phosphate group. So we add a phosphate group, and that traps it to form glucose 6-phosphate. The 6 just denotes what carbon the phosphate group is on. And we do this by taking a molecule of ATP and forming ADP, adenosine triphosphate to diphosphate, and we take one of those phosphate groups, which requires energy, to trap it inside of the cell. Now, let's just think back to general biochemistry. What enzymes add a phosphate group or transfer a phosphate group? Well, yeah, that's going to be a kinase. So if we remember that glucose is a 6-carbon molecule and we add a phosphate or we transfer a phosphate, we could remember that this enzyme is hexokinase. One note is that in the liver, a similar enzyme called glucokinase does the exact same job. Next, glucose 6-phosphate then gets isomerized into fructose 6-phosphate. This arranges the molecule from an aldose with an aldehyde group to a ketose sugar which contains a ketone. More on that in a future video. For this depiction, they look the same because we have the same amount of carbons and the phosphate in the same spot, but the actual structure of the molecule is different. So what type of enzyme isomerizes? Well, obviously it's going to be an isomerase. So if we have a glucose that is phosphorylated, that is then isomerized, it would make sense to call our enzyme phosphoglucose isomerase, and that's what we call it. So now take a look at this arrow. One big reminder is that the steps that require ATP are generally going to be irreversible reactions, such as hexokinase up here. This isomerization is a reversible reaction, and the enzymes can work in either direction. So now our last step, step 3 in phase 1, is to add another phosphate from ATP to the first carbon. When we add another phosphate, we call this a bisphosphate. So a fructose molecule with a phosphate on carbons 1 and carbon 6 is called fructose 1,6-bisphosphate. So this is another irreversible reaction because we require an ATP molecule. So think back, what type of enzyme is going to be responsible for transferring a phosphate group onto fructose 6-phosphate? Well, if it's a fructose molecule with a phosphate and we know it's a kinase, we can remember that this enzyme is called phosphofructokinase 1. Now, there's a phosphofructokinase 2, but that is for a separate reaction. PFK1 is actually, interestingly, going to be our major control point and rate-limiting step of glycolysis. Normally in biochemistry, our first step, such as hexokinase, is going to be rate-limiting. But glucose 6-phosphate can be in the cell from other pathways, such as glycogenolysis. So we have to have a rate-limiting step that is after the production of glucose 6-phosphate. In this case, PFK1 takes that job. So here's your general roadmap for phase 1. Okay, so we just walked through phase 1, the investment phase. Just to overview, we took glucose, and then we phosphorylated it to form glucose 6-phosphate, isomerized it to fructose 6-phosphate, and then we added one last phosphate to get to fructose 1,6-bisphosphate. So now let's talk about this middle phase, cleavage. Phase 2 is where we have the great split. We split from a 6-carbon molecule into 2 3-carbon molecules. From here on, we get 2 of every intermediate and other products that we produce. Okay, so again, phase 2 is where we are splitting from one 6-carbon molecule to 2 distinct 3-carbon molecules. The big overview is we start with our fructose 1,6-bisphosphate, which we created from our investment phase, and we will create 2 glyceraldehyde 3-phosphate molecules. Our first step during this phase is simply cleaving fructose 1,6-bisphosphate into 2 distinct molecules. One is going to be glyceraldehyde 3-phosphate, and the other is going to be dihydroxyacetone phosphate. Because we have an enzyme cleaving this molecule in half, we only need one enzyme in order to produce these 2 products. So we move from a 6-carbon molecule with 2 phosphates into 2 3-carbon molecules, each with 1 phosphate. So this enzyme is fairly special in its name, so it is unfortunately just memorization here. This enzyme is called aldolase, which again splits fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. One interesting note is that dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are isomers. This is a similar process where we move from a ketose to an aldose sugar. If we put it together, we have a 3-carbon molecule that is a sugar, and this has 1 phosphate, and it is being isomerized. So we can remember that this enzyme that does this interconversion between the two is called triose phosphate isomerase. So which one of these molecules is more important? Well, it's going to be glyceraldehyde 3-phosphate. This will go through and finish the rest of glycolysis for us. Also, a quick note, if you look at these arrows, all of these reactions are reversible. So these are all your reactions for phase 2, the cleavage. So we have taken this time to invest 2 ATP into this process. Then we chop the end product of that in half to form 2 products, which then isomerize to form 2 glyceraldehyde 3-phosphate molecules. It's time to get some payoff or profit from all of the work that we have put in. In this phase, not only will we generate some energy, but we will also create 2 molecules that can go to the Krebs cycle to generate even more energy for us. Phase 3 is when all of this happens. We go from a 3-carbon molecule of glyceraldehyde 3-phosphate to another 3-carbon molecule, pyruvate. So we know we are not cleaving or losing any carbons along the way, but we are changing this phosphate molecule. In this process, we create ATP and NADH as our main products and also some water as a byproduct. So here's the more in-depth process that we will walk through step by step. We will begin with glyceraldehyde 3-phosphate. This then becomes 1,3-bis-phosphoglycerate. This process is done by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. And this enzyme is special because it is the only dehydrogenase in all of glycolysis. So let's take a look at what's going on here. We have two 3-carbon molecules with one phosphate attached, which then goes to two 3-carbon molecules with two phosphates attached. So keep that in mind. We're adding a phosphate group. So let's take a step back. Now let's talk about what do dehydrogenases do? Well, a dehydrogenase is a special type of oxidoreductase, which is a type of enzyme that catalyzes the transfer of electrons to an electron carrier, such as FAD or NAD+. So in this case, glyceraldehyde 3-phosphate dehydrogenase catalyzes the formation of NADH from NAD+. Now, the interesting note is that instead of an ATP, this reaction uses an inorganic phosphate denoted as this PI. So even though we added a phosphate, we did not use a kinase or an ATP. Remember that this is the only exception in glycolysis. So now we move towards our ATP generating steps. Remember that at this point we are at 3 carbons for the remainder of glycolysis. Our first step involves 1,3-bis-phosphoglycerate, or two of them. This forms 3-phosphoglycerate by losing one phosphate group on carbon 1. Where does this phosphate group even go? Well, interestingly, it goes to ADP in order to form ATP. This is what we call substrate level phosphorylation, or SLP. Substrate level phosphorylation synthesizes ATP by the direct transfer of a phosphate group to ADP. Now, this is different from oxidative phosphorylation, which requires the electron transport chain and ATP synthase. So remember, I will continue to harp on this. What enzyme transfers phosphate groups? Yeah, again, it's a kinase. So because this is phosphoglycerate and a kinase, we simply call this enzyme phosphoglycerate kinase. The next step that happens is we change where the phosphate group is. So again, we keep the same carbon structure, 3 carbons, but we move where our single phosphate molecule is. I like to think of this as a mutation because it helps with the enzyme name. The phosphate moves from the 3rd carbon, where it is right now, to the 2nd carbon. So instead of calling this 3-phosphoglycerate, we call it 2-phosphoglycerate. Because this is a phosphoglycerate molecule and it mutates from 3-phosphoglycerate to 2-phosphoglycerate, we're going to call this enzyme phosphoglycerate mutase. In the second to last step, we create a molecule or an intermediate called phosphoenolpyruvate from 2-phosphoglycerate. This enzyme that does this is called enolase. Phosphoenolpyruvate, or PEP, has a double bond in it. And in order to form this double bond, enolase removes a molecule of water. So we generate 2 molecules of water because remember, everything is doubled. So our final step in glycolysis is going to be the formation of pyruvate from phosphoenolpyruvate. This is finally our final product. During this process, we undergo another substrate level phosphorylation. So what else do we make besides pyruvate? Yeah, we form 2 ATP from 2 ADP. And what enzyme do you think does this? Well, this follows our same pattern. So if we know that we're generating pyruvate and we're transferring a phosphate molecule, we require a kinase, so we can call this pyruvate kinase. So that is all of glycolysis, and this is the end of phase 3. With all of that, what about some final accounting? So what did we use during our investment phase? Well, we started off with 1 glucose, and we also used 2 ATP during the steps of hexokinase and phosphofructokinase 1. Did we use anything during the cleavage phase? Well, no, we didn't, so I'll just put Na for now. What about our payoff phase? What did we produce? Well, we made 4 ATP, we made 2 NADH, we made 2 H2O, or water molecules, and 2 pyruvate molecules. The ATP were generated by phosphoglycerate kinase and pyruvate kinase via what mechanism? Yeah, substrate level phosphorylation. And the NADH was generated by glyceraldehyde 3-phosphate dehydrogenase. And lastly, the water was generated by enolase. So that's glycolysis. Even though this is a simplified graph without all the actual molecules, it is a useful framework for thinking, visualizing, and understanding glycolysis. Take time to walk through the entire mechanism yourself and think through all of these steps. I know we went through a lot of information really quickly. Rewatching this video and actively engaging with the material will help to continue solidifying this. If you are able to track the carbons and phosphates, you aren't just memorizing the intermediates and enzymes, but are truly understanding what is happening in glycolysis. My biggest tip is being able to write out all of the steps of glycolysis over and over again, but truly understanding each step and each enzyme and what they produce. I hope all of this made sense, and I hope it all helped. Thanks for watching.